Hello. It's the
Hi.
second time we've been here this week. I don't know that we've done two events in one week, but we're really happy to be back again.
Yeah, you didn't even have time to miss us.
I know, but welcome to everybody who is joining us. And this time it's morning on the West Coast. We don't usually do that, so I hope you have a cup of coffee if it's morning for you, and maybe you have your lunch if it's the afternoon on the East Coast. But my name is Anne Merchant, and I work for the National Academy of Sciences, and I am joined by, as always.
Rick Lovard. I also work for the National Academy of Sciences. I'm the program director of the Science and Entertainment Exchange.
And of course, your background is very NAS today.
That's right. That's right. I'm sitting in the boardroom at the National Academy of Sciences, maybe.
Yes, maybe in a virtual kind of way. Exactly. So we
Oh,
we
by the
start
way, photo credit and merchant.
well. And we like to say that the portrait behind you, our famous Lincoln portrait, is one of the first examples of a version of Photoshop because, as I understand it, Abraham Lincoln was not available to sit or stand, as it were, for that portrait, and had to be painted in after the fact. So it's a little bit of 1800s photoshopping. So that's kind of some fun trivia about that portrait. But I guess I should start, as I always do, by explaining a little bit about what the National Academy of Sciences is, because, when you say national and you know that we're sitting on Constitution Avenue in Washington, D.C., it sounds very much like we're part of the federal government. But Abraham Lincoln, and of course, the reason that portrait includes Abraham Lincoln, was the president at the time, as we like to say, in the travail of the Civil War, that signed the National Academy of Sciences into existence through executive order, and wanted an institution that sat outside the framework of government to guide good federal policy, and often state and local policy, that is informed by expertise that comes from science. Now, the National Academy of Sciences is joined by sister organizations over the years that now include the National Academy of Engineering and the National Academy of Medicine. So we are very happy, for example, there are members of the National Academy of Sciences, and it's a very rarefied group. There are just about 2,000 of them. And of course, Jennifer Doudna, who is joining us today, is one of those members of the National Academy of Sciences. So we're always super happy when we can get an NAS member on our stage, either virtual or in real life. So that's a wonderful thing for us to be able to say at the top of an event. And so the Academy does a lot of work in a lot of different areas. I mean, it's a huge swath of science, engineering, and medicine. But I think relating to today's event, we convened two international summits on human genome editing, the first of which was in Washington, D.C. in 2015, and we followed up in 2018, late 2018, with a second international summit that was in Hong Kong. And I think that one made the news because it was at that summit, just prior to, I think, the opening to the summit, that the Chinese scientist announced that he had actually created two gene-edited children. So this is important work. The Academy does this to sort of help frame the context for the governance and the ethical constructs that we need to think about with a tool as powerful as Crispr. But of course, we hope that you've all watched this film, Human Nature, and that you have a good sense of what this means and why we think about this, because it's a really powerful way to tell science stories that are so important. Not everyone's reading the 400-page reports that come from the national academies, and we understand that, but we hope that you're watching films like Human Nature. And of course, Adam Bolt is here with us today to talk about the art of science storytelling. But I do want to make sure that, Rick, that you explain why the National Academy of Sciences has this connection to Hollywood through the Science and Entertainment Exchange.
Yeah, so if you're a writer, producer, studio executive, actor, director, storyteller, really of any kind, and you're making a mass media project, like a film, a TV show, a video game, you have a question about science and you want to put it in context, you really want to have a conversation over about it. You don't just, want to go on Wikipedia and try to find the answer. You can call 844-NEED-SCI, and either I or Sachi will pick up the phone. You can also email us, of course. And we've done over 300 of this type of events to try to bring entertainers and filmmakers together. And we've done over 3,200 consults, including Avengers Infinity War, Man of Steel, Black Panther, Captain Marvel, more recently Songbird. And on our last event, documentary filmmaker, reminded us that we also do work on documentaries. Film Food Evolution by Scott Kennedy. Now, I'm not going to tell you who reminded us that we do work with documentaries, but also we work with Scott Kennedy. So
yes.
If you are a STEM professional and you have a question and you would like to do this type of consult and volunteer, then please would like to hear from you. So please do email us or be in touch or dial the number that Sachi put up. I want to thank a couple people really quickly. Ameche for doing our AV and making this whole thing run smoothly. We couldn't do it without you. Thank you. Also, the office in DC, Courtney Sloan and Jeff Fishman, and my colleague in LA, Sachi Gerben. Also, our sponsors, Howard Hughes Medical Institute, who sponsors our event series. So it's, this today, Howard Hughes, thank you. Also, the Alfred P. Sloan Foundation, Walt Disney Company, Film Nation, Google, the Lyda Hill Foundation, Corteva, and many individual donors like you. We had a lot of people this time around who gave at the supporter level. You will be getting a challenge coin from us and also unique access to our events. Thank you so much. Any donation helps, but for those who can join at the supporter level, we really appreciate it. It really goes right back into sort of the production value on these events and making more and better events. And so today, Walter, our moderator, will be speaking with Jennifer and Adam. And if at any point in that conversation, you have a question, you can enter it in the little bubble that says Q&A that's either at the top or the bottom of your screen. And we will be taking as many questions as we can. There are always more questions than we have time for in the audience Q&A, and we will be feeding them back to Walter. We apologize if we don't get to your question today. It's most definitely my fault and not Walter's if we didn't get to your question, because I'm going to be the one sending him the questions. So I apologize in advance. We'll get to as many questions as we can.
Yeah, and this promises to be a really good event. We're excited to have this panel. So we always start with a couple of little rabbit hole things. I wanted to refer you to an article in the New York Times. It's about a group called Kids of Survival. And this is relevant to the Academy because in our West Rotunda, we have a work of art that hangs there that relates to evolution and the origin of species from Darwin, along with our Darwin bust that's in there. So Kids of Survival was a group that was created in the 1980s by the artist and educator, and I'm looking down to find his name so I get this right, Tim Rollins, who worked with a group of students, at-risk students from the Bronx that were learning disabled, but tried to encourage them to connect to education through the arts and specifically to science. And those kids all went on to become actually very successful and remain highly connected to the arts. And our cultural programs of the National Academy of Sciences continues to work with them and will be doing more with them, and you can probably catch up with them through the cultural programs. But I really encourage you to read that article in the New York Times because it's really interesting and very inspiring.
All right, my rabbit hole item for this event is, so my sister gave me this complete score of the Beatles, every song they ever recorded, every note they ever played, and it is fantastic. It's a Hal Leonard book. If you are a musician and you play any instrument that the Beatles played, it's in here. I learned in my life this week. It's a great book.
So next week you're going to be serenading us. I can see what's coming. I can see it.
I don't know. I could be swayed. I don't know. Maybe.
Well, we're going to now turn this over to Walter Isaacson, which is, I'm sure the name is very familiar to you, as will be the face. Of course, Walter is a well-known biographer and, in fact, has written a biography of Jennifer Doudna and is a professor of American history at Tulane. And we're very excited that he is here today to moderate the conversation between these two incredibly interesting people. And so we're not going to take any more time from you, Walter. We're going to hand it over and allow you to have our virtual stage.
Thank you very much, Anne, and thank you very much, Rick, and thanks to the National Academy for all you do, and especially for the convenings and the international summits that you've done around the issue of germline gene editing and CRISPR. You've certainly been in the thick of this discussion, which I hope will continue during this session here. We're in the third great scientific revolution of modern times. The first was the first half of the 20th century, And it sprang from a lot of it sprang from Einstein's theories in 1905 on quantum mechanics and general relativity, which leads us to things like the transistor and space travel and microchips, the atom bomb. It was a revolution in physics. The second half of the 20th century was dominated by an information technology revolution that came really from three inventions: the invention of the microchip, the internet, and the computer. And when those were combined, we had a digital revolution that for the second half of the 20th century was the technology innovation engine of that period. We're now entering a third great revolution, and that's a revolution based on life sciences. In some ways, it begins in the early 1950s with the discovery of the structure of DNA, but it is really kicked off around the year 2000 when the Human Genome Project starts publishing the human genome. And then people like Jennifer Doudna and others discover it's not only the DNA, but the RNA that matters, and how RNA can be a wonderful tool. We've seen it become a wonderful tool with the coronavirus. I'm a member of I'm a participant in the clinical trial for the Pfizer vaccine, and partly I did so because I was very eager to understand better the role of messenger RNA. But what Jennifer did starting in around 2008, 2010, was look at a biological system that bacteria have for fending off viruses, something we could certainly learn to use these days as we fight off viruses. And that system used an RNA-guided enzyme that acted as a scissors to help edit, cut up the DNA of an invading virus, invading the bacteria. But Jennifer, Emmanuel Charpentier, they won the Nobel Prize for their discovery of exactly what were the components of this system and how it could be used to edit the DNA of a living organism. And so with that, I want to bring Jennifer on stage, ask her to turn on her video, and then also bring on Adam Bolt, because as I was researching this story, I saw the film Human Nature. I love films, I like documentaries, but I wasn't thinking too much of it. But about 2 minutes into it, I realized that this was something absolutely special. It addressed all of the complexities of genetic editing in CRISPR, all of the human factors involved, but didn't shy away from the science. And yet it was absolutely riveting. Jennifer is one of the stars of the documentary. And so I really recommend that you watch it. And before we fully begin, I want to begin with a question to Jennifer, but actually I'm going to turn to Adam because Amita Masoor has already posted a question, which is, how can I watch this documentary? So let me let you start, Adam, by telling people how, if they haven't seen it yet, they can watch this documentary.
Yeah, well, as of this fall, we're up on Netflix, so that's probably the easiest way for a lot of people. And another way, if you don't have access to Netflix, the film is available on some library platforms that people may have access to through university or college, so you can find it there.
Jennifer, congratulations on the Nobel Prize. I was wondering if you could explain to us how you got interested in CRISPR, what it is, and then recently how you tied it in to the whole notion of us fighting COVID-19.
Well, hi, Walter. It's great to be here with you and Adam. CRISPR is a wonderful story of fundamental science, small science that went in an unexpected direction and led to this extraordinary technology for changing the DNA precisely in cells or organisms. That's fundamentally what CRISPR is as a tool for scientists. And I'm sure we'll get into some of the really interesting applications of that tool, and that's something the film Human Nature gets into as well. But I want to point out that in biology, CRISPR is an adaptive immune system in bacteria. It's a way that bacteria find and fight viruses, something seems very relevant in our current pandemic situation. And it was by studying how that works, the molecules that are involved in that immune system, and in particular, a protein called CRISPR-Cas9, and its ability to be programmed to find and destroy viruses, that Emmanuel Charpentier and I and our colleagues were able to figure out how to harness the system as a tool for altering the sequence of DNA in cells.
And how have you used it to help fight the COVID pandemic we're in?
Right now, there's a very active effort in a number of academic labs as well as companies to use CRISPR in a mode that is more reminiscent of what it does in nature, namely finding and then reporting on the presence of viral genetic material. So the virus that causes COVID-19 is a virus whose genetic material is RNA. It's a chemical cousin of DNA. And the CRISPR system, some forms of it, are very good at finding those RNA sequences of coronavirus and signaling their presence. And so this is done in laboratories by using CRISPR in a mode where it can actually turn on a fluorescent signal when it encounters and detects the coronavirus. And this is how it's being used right now as a diagnostic tool.
And you have a company that you helped found called Mammoth, and it might have home detection tests using CRISPR within this year. Is that possible?
I think it's possible. It's certainly a challenge, that's for sure. I think that one of the things to keep in mind about CRISPR is that it's a technology that, because of its programmable nature, it will be useful not only for the current pandemic, but also for future detection of other viruses, including influenza, but other forms. It's easily programmed to find new variants of the coronavirus that we continually read about in the media. So I'm hopeful that it will be available to address the current pandemic, but certainly for future preparedness as well.
It's interesting to talk about it being programmable, because in some ways in this new life sciences revolution, molecules are the new microchips. And the thing that you've been able to do, both with the RNA detection, but also the RNA guided enzymes that are used to edit our DNA, is that they can be pretty easily reprogrammed just using the 4 or 5 letters of RNA and DNA, as opposed to the 0s and 1s of a microchip. Adam,
exactly.
Adam, when you did this movie, you had to make a lot of choices about how you were going to make it both personal but also scientific. Tell me about the choices that you made in making such a documentary.
Well, I mean, it felt like an enormous responsibility in a way. I really loved the way you framed this whole conversation, Walter, talking about biotechnology as a third great scientific revolution. And, it kind of felt like this was an opportunity to document this formative technology in that revolution, almost like being there when the transistor was invented or during the personal computer revolution. So, partly we looked at this as kind of like an oral history of this amazing discovery. There were a few things, this being such a major discovery that, even when we started working on the film 5 years ago, was clear that this was going to be foundational and having ripple effects on science for many years to come. We thought this was something ordinary people deserve to actually understand. So that was an early goal of the film, is to really explain some of the nitty gritty to people. Some of the stuff Jennifer just explained so well. We wanted people to understand that, and we wanted them to understand some really kind of bigger picture things about this discovery, which both of you touched on. One thing that struck me early on was that, a lot of people have this image of scientists, especially if you talk about something like genetic engineering, that maybe there's almost like the mad scientist cliche, like there's these scientists kind of in their labs in the dark, figuring out what crazy Pandora's box they can open up next. And that wasn't the case with this discovery at all. And that was very interesting to see how this was something that unfolded over decades with lots of contributions from different scientists who were really just trying to understand how the world worked. So we wanted people to understand that it came out of this basic science. And then we wanted them to understand sort of what you just said, Walter, that this is a revolution driven by molecules, and these molecules, at least the basic parts of them are coming from nature. So we wanted people to understand that this is a technology that actually relies on billions of years of evolution that have created these components, which I think kind of shifts your idea of how much do humans know versus what we're kind of building on from nature, and what kind of humility maybe we should have as we go toward using this technology. And then we wanted to really give people a lot of different perspectives on the ethical issues, which immediately come up once you start talking about the possibility of changing this most fundamental part of our being, and possibly altering evolution, really.
Well, Adam has talked about the ethical issues. Jennifer, I think there was a nightmare you had at one point that started you down the path of joining forces with the National Academy of Sciences and so many other people, David Baltimore and others, to say, I've explored the science, but now it's time for me to explore the ethical issues as well. Tell me about that progress.
Very early in our work on this technology, it was clear that it was a truly extraordinary tool, in the sense that, as Adam just said, it can be used to change the code of life, to change fundamentally the instruction set that cells have to make us who we are, or any other biological system on the planet. And so I started, really thinking about how, in a way, profound that was, and what kind of opportunities, but also risks, kind of lay ahead with a technology like this. And so I did have a dream one night that I still remember quite vividly, about being introduced to a person in a darkened room. And when that person turned around in his chair, it was Hitler. And he had sort of a pig nose. So it was almost sort of already representing kind of the, sort of horrifying possibilities of, the extremes of genetic engineering. And this, in my dream, this figure asked me to explain CRISPR to him. And I realized, sort of with a horrifying, start, that this was someone who had ill intentions in mind with this technology. And so when I thought about it later, it really was one of the things that kind of pushed me to begin the process of discussing the technology much more broadly, encouraging transparency among the scientific community. And I was very pleased that the National Academy stepped up early on to organize the summit in 2015 that has then led to a second summit. And I think there's a third one planned later this year in London. And so, this has really been an opportunity, I think, for scientists globally to come together and make a very strong statement about how this technology should, and frankly shouldn't, be used in the near term.
At that second summit that you just referenced, the one in Hong Kong, it's when the Chinese scientist He Zhuangqi announced that he had edited the embryos of two twins to make them not have the receptor for the virus that causes AIDS. You were there and very much a part of having him speak. I see in the chat room that we have Valeria asked, were we ever going to know more about those Chinese babies? So I'll put that to you. But I also put to you the question of what did you feel and what did you think when you heard that what you had invented, had discovered, had been used to do germline editing. And by the way, now that we're fighting off receptors for viruses, has your thinking changed a little bit from your initial horror at what Homogeneity had done at that international summit?
Well, first of all, it certainly was horrifying to receive an email from Homogeneity. It was Thanksgiving weekend of that year, and literally right before I was scheduled to depart for Hong Kong for the summit. And you know, I think the subject line was "Babies Born," and it was you know, a very short email just announcing what he had done and his intention to publicize this. And I think, you know, this of course kicked off an extraordinary scramble with the National Academy team and all of us who were organizers of the conference to figure out how to manage the meeting with this, you know, this incredible you know, news that was coming out, and also how to try to understand this person and this effort. Why had he done this? Why did he think this was okay to do? And so that was one, you know, sort of set of events. But I think, you know, your second question about have my views changed, I think that a couple of things have really, you know, in particular, I guess, have really affected my views about what we refer to as germline editing. So that means making genetic changes in sperm or eggs or embryos. And if you do it in, of course, of human beings with the intention of, you know, if you create a pregnancy, then those changes are heritable. So they can be passed on to future generations. I think the reality is that this is certainly coming at some point. I don't think we can, you know, you think that we can sort of prevent that type of work from being done ever in the future. However, I do think that there's a very, very important set of guidelines in place right now for future plans of that nature that has been put forward now by multiple groups, but including the National Academy of Sciences. And also, I think that frankly, there may be opportunities in the future to use, you know, germline editing in ways that will be beneficial to people. And we may get into that when we talk a bit more about the film. So I'm open to that, but I think it has to be done with extreme caution.
Adam, one of the ways you address this in the film is through a very young but very wise philosopher named David Sanchez. I joke about him being a philosopher. He's a kid in high school who has sickle cell anemia, loves playing basketball, except for when the pain makes him double over. And I was wondering if you could describe David Sanchez, just turned 18, how he is these days, for those of us who've watched the film, but also the moral issue that he raised when you asked him the questions.
Yeah, David, our producer Meredith just talked to him the other day, and his grandmother. He's doing well. He is in his senior year of high school, of course, attending remotely like so many kids are. Still getting those regular apheresis treatments that we show in the film, where he has to have this major transfusion of healthy blood. He has not had a sickle cell crisis in a while. So he's doing well. Yeah, that moment in the film was kind of a total surprise to us. You know, we wanted to. I mean, it was really important to have David in the film as someone who could, you know, speak to the perspective of the people who this work, you know, a lot of it is being done in their name to cure, prevent these horrible diseases. And we really approached it like David was an expert on his own condition, as, you know, people who live with a serious disease are. You know, they have to learn the science and the treatment options and all that. And he was incredibly aware of all that stuff. But it kind of on a bit of a whim, I just threw at him, what did he think about the possibility of germline editing for sickle cell disease to possibly prevent, if he had children in the future, or just, hypothetically, other people out there using it to prevent their children from having sickle cell disease. What did he think of that? And his response in part was, he very thoughtfully kind of paused and considered it and said, shouldn't it be up to the kids? I don't think I would be me without sickle cell. And I think that was really striking to me, because he put his finger on an issue that is also very much in discussion among academic bioethicists who have studied these kinds of issues their whole career, which is this kind of paradox about changing something so fundamental about who you are, even if it's something bad, something that has lots of negative consequences, but it might change other things about your life that you might regret. And in David's case, he talked about the patience and empathy and humility that he's learned living with this disease. Now, I want people to know, David isn't saying this shouldn't be out there. And I know, Walter, you talked to him also, so you could add to that. But we kind of sprung this question on him, and he immediately put his finger on one of the paradoxes of germline editing. And I think it was important to include in the film, just so that people don't take for granted that, they don't take for granted anything about what people who might benefit from this technology actually think. And David's one person, so other people would have different views. And I think David's view is actually, it's more nuanced than just yes or no. I think he was just pointing out some of the paradoxes, but it's really important for those people to have a voice in this.
You're right. It was an incredibly powerful part of your great documentary, which, when people get to it, they will love that part. And I did follow up at posing some questions to him for the book that I have coming out in March on Jennifer, and called The Codebreaker. And I wanted him to wrestle with the notion of, well, would you want your children to have sickle cell? And of course, he says, well, maybe if you could cure it, I wouldn't want my children to have sickle cell. And that's part of a whole section in the book, which I'd like us all to discuss, which involves what should we allow parents to do for their children. Maybe preventing them from having Huntington's disease or sickle cell or cystic fibrosis or simple genetic problems, those being fixed. That seems logical, but what about if we could enhance their height or their muscles, or even enhance their IQ? Is that something we would want? I know your colleague and close friend, Jennifer, George Church, says in my book, well, what would be wrong with allowing people to buy more IQ points for their children? Wouldn't that be a good thing? But to me, there are a couple of moral issues that come up when we start allowing people to buy better genes for their children. I mean, Jennifer, you've wrestled with that. What do you think?
Well, I think first and foremost, it's very important for this technology, frankly, any technology, but we're talking about CRISPR here, to not exacerbate the already existing problems of, inequities across the planet. And that sounds to me like not the thing we want to be doing, is, making it possible for the wealthy few to tweak their children's genes in ways that might give them, some kind of additional advantages to what they already have. I personally feel that the most important, certainly in my own work right now, the thing I think about all the time right now is, how do we make CRISPR affordable and accessible to people that need it? And I'm not talking about germline editing here. This is just, thinking about, even the example of sickle cell. How do we make sure that everybody that wants this treatment for sickle cell can get access to it, and not just in the US, but globally? And that's right there is a very big challenge, because currently the cost of that treatment is north of $1 million a patient. So, you know, I think there's some things scientifically and technically that can be done to help address that. But anyway, you know, I really strongly feel that we don't want to go in a direction that exacerbates existing inequities across the world.
You talk about using CRISPR to help cure sickle cell. That actually has been done in this past year using CRISPR-Cas9 technology that you and Emmanuel Charpentier pioneered. Tell us about how that works and what you and Fyodor Urnov and others at your lab are doing to make sure that that can be used as a normal course of medicine at some point and not be prohibitively expensive.
One of the earliest set of clinical trials using CRISPR has been to address sickle cell disease. And there have been several announcements about the ongoing outcomes of those trials. And probably the most exciting was the news that Victoria Gray, the first patient to be treated in the US for her sickle cell disease using CRISPR, is continuing to do very well. And she's, I think, now more than a year into her treatment. The CRISPR technology that's being used in her case and in a few other patients like her is being used in a way that doesn't actually correct the disease-causing mutation, but what it does is turns on production of fetal hemoglobin, which can suppress the defect in the adult hemoglobin gene. And so that's certainly a strategy that is clearly working for these patients. It's very exciting. I think one thing to note, though, is that in these ongoing trials, the negative effects have all come from the bone marrow transplantation process that is part of this therapy right now. So in our own labs at the Innovative Genomics Institute in the Bay Area, and with my colleague Fyodor Urnov, who plays a prominent role also in the Human Nature film, we are hard at work at some kind of next generation strategies that will not require bone marrow transplantation for treatment. Imagine a day when we could have a single injection, or maybe at some point a single pill that a patient can take, and that is sufficient to target the cells in the body, in the bone marrow, that need editing, not bother any other cells, and lead to an effective cure. And it sounds a bit science fiction-y, but I feel strongly that we can get there by focusing on the technical aspects of that.
Adam, you filmed part of this in Turkey in an archaeological dig, and it really puts a wonderful frame on your documentary. Explain why you did that.
Well, I mean, going back to what you said earlier about this being, one of the major scientific revolutions of this century, we're just at the beginning of it, and from the beginning of making this film, our team was thinking about where is this technology going to take us in 50 years, 100 years, and if you start thinking beyond that, it really becomes kind of mind-boggling. So, we tried different ways to try to peer into that uncertain future. Some of that was considering what science fiction has to say about this. I was a big fan of Gattaca as a teenager, so we look at that. We look at Jurassic Park, different things that have tried to play out long-term possibilities of what genetic engineering could do. But we also thought, why not look at the past and think about, in the case of going to Turkey, it was looking at a technology that a lot of us don't think of as a technology, which is agriculture and selective breeding, which came on the scene 10,000 plus years ago. And so we went to a place in Turkey called Çatalhöyük, which is one of the earliest sites where those selective breeding and agricultural techniques were seen, and spoke to an archaeologist there, kind of asking the question, if these people were scientists, what what could they see and what could they not see about where that very early technology that you could almost think of as a kind of genetic engineering, where that would take society. And I think, it gives you a really big frame to think about where we might go with CRISPR, because of course, agriculture is fundamental to human civilization at this point. It's so fundamental that we think of all of its products, the fruits and vegetables that are at the grocery store are natural products, even though if you look at something like corn, and then you see this wild grass species called teosinte that was the progenitor of modern corn. Corn looks like a yellow vegetable Frankenstein, kind of compared to where it came from. But that's natural now. And it's how we're able to have such a large population and feed them, but it's also, there's all kinds of environmental costs to that. It's transformed the surface of the planet has been given over to agriculture. So I don't know that there's not any kind of specific answer to what CRISPR will bring on that kind of timescale, but I think we just wanted to put the audience in the mindset of realizing that these things have ripple effects over decades and centuries and millennia.
Let me go to some of the questions that are great questions that are popping up in the Q&A room. And Geetha asked, and this is for Jennifer, if you use CRISPR-Cas9 to undo a gene or to edit a gene, can you reverse the process if something goes wrong?
Well, in principle, the answer is yes. It might not be possible to make an exact chemical reversion of the edit, but yes, in principle, it's the editor could be used to edit your edits if you wanted to take it to that level.
And of course, there's always the question, Adam, in discussions like this, which is, I'm a young scientist and a filmmaker. What would you advise me to do in order to communicate science better? How did you become a filmmaker? What advice do you have?
Well, I mean, I don't know if this holds in pursuing science, but certainly in pursuing film, the best advice I can give is just to make things and experiment and, kind of try things out and see how they go. In terms of communicating science specifically, there's a kind of a perception maybe out there that scientists themselves aren't the best messengers for communicating the complex research that they're involved in. I really did not find that to be true. All the people I talked to were amazing communicators, but I think that the key to it was building trust in terms of making sure that the folks that I was talking to knew that I'd sort of done my homework, and that, I think rightly so, scientists can be worried that they're going to be interviewed for something and then that what they say is going to get taken out of context and sensationalized. So, it was important to make sure that the scientists knew that we weren't going to do that. And it was also, I tried to kind of put the scientists into teacher mode a little bit. I think a lot of these folks are, in addition to being amazing researchers, they teach and they're amazing teachers. And if you can kind of approach them with that mindset of really trying to make the film a way to teach the audience, and I just tried to put myself in the audience's shoes and think about things that maybe were confusing to me, even if I eventually understood them after months of research and talking to different people. I was sitting in the interviewer's chair. I tried to play the part of the audience member who didn't understand that thing yet, to make sure that, whatever jargon and stuff that might get in there would get demystified.
Well, you're being a little bit modest too, because what you do is you make it into a narrative, and you're a great storyteller. And I do think that the key to anything like this is, let me tell you a story. So congratulations on you, how you made it such a storyteller. Let me get the names here. Cassidy and others, and Estefania, what measures should government be taking to regulate who can have access to this tool, and how can you ensure that other countries, you wouldn't have, say, medical tourism, where other countries would not regulate the access as strongly? Jennifer?
Yeah, these are big challenges, and they're not unique to CRISPR. They're challenges that we have for any new technology in the biological sciences or beyond. In the case of CRISPR, we're fortunate that back in the 1970s, scientists had to grapple with the ethics and sort of the ramifications of molecular cloning, and that led to a set of guidelines that were instituted at the National Institutes of Health, which is the agency in the US that handles a lot of government-funded, taxpayer-funded fundamental research, medical research. And so the use of CRISPR really does come under those guidelines very, very well. I think that it's a challenge to have government regulation of any technology. I mean, we're seeing this playing out with technologies that are involved with social media right now. We see a lot about that, but again, it pertains to other things too, where we can't really expect our government officials to be experts in all of these different areas, and these are complex technologies. And that's why I've really advocated having the scientific community be actively involved in the case of CRISPR. And of course, the national academies play a very important role there in convening the right groups of experts that can weigh in. And then in terms of access and the possibility of medical tourism, it's very hard, I think, to guard against that completely. I do think that, our best hope really is to have, as much transparency as possible about where and how CRISPR technologies are being used, especially for things like germline editing in humans, and to just continue to have these international summits where people can come together and discuss the technology and the science, which is moving very, very quickly, as you probably know, but also the broader impacts, and have other stakeholders at these meetings that can speak to those impacts.
There's a question from Dawson, which I guess also goes to you, Jennifer, and it's a question I wrestle with in the book, that in addition to the inequities that could come from gene editing, there's also the question of the lack of diversity in our species that could be the result over time of genetic editing. And Dawson asked, can you speak to the possible consequences of the increased genetic homogeneity, both for the genome and for culture, and I would say, and for the species?
Right. Well, and again, that's really a question that takes the long view, right? Because even if we started doing that today, it would take many generations to have, to see real, changes broadly across our species with CRISPR. But I think you raise a really interesting point that I think about, again, taking the, what's going to happen, as Adam was saying, this is not a technology that's going to have an impact for a year or two. It's going to have an impact over decades, centuries. It's hard to predict beyond that. And so we do have to be thinking about, these kinds of implications, especially as scientists are, continuing to advance the tools, and then practitioners are, quickly adapting them for, various uses. And, in the short term, just to take the short-term view for a moment, I think the technology isn't at a point where it could be widely utilized for human germline editing for multiple reasons, even if one wanted to. But again, thinking ahead to what's coming, I do think as the technology continues to improve and our knowledge of the human genome continues to advance, it'll become increasingly practical to do this kind of germline editing. And then these questions really do come to the fore. So I'm just, continuing to advocate, the active engagement of the scientific and clinical communities to discuss these issues.
Adam, that leads to one of the questions in here too, which is the active engagement, not only of the scientific community, but all of us as citizens, as neighbors, that we shouldn't go marching down this path. Many people have said, there's a book Francois Baylis just came out with saying it, unless we have a broad societal discussion of this issue. To what extent does what you do, this notion of a film like this, is it geared to help spur a broad social discussion of the issues?
I mean, that's certainly our hope, to be part of that process. And that's part of what I love about the cinematic experience, which unfortunately is very much changed right now. But, the idea of bringing people together in a dark theater to kind of watch something and experience and react together, and hopefully to have some kind of Q&A afterwards or something for people to discuss it. But even, in just people seeing the film and learning more about this however they can, I think that's key. One of the great subjects in our film, Alta Charo, who's a bioethicist, talks about, this idea that it's not the technology itself isn't, good or bad. It's the user, the person who decides how to use it. And ultimately, that's going to be all the people in the audience and all the people across the world who, have health issues, are considering planning a family, and genetics becomes part of that. And I think it's really important that that they have a nuanced understanding of what this technology can do, as opposed to just, there's the kind of cliche idea of raising your kids' IQ points by buying a gene that'll do that. But when you dig into the science of that, it's not nearly that simple. I mean, we could have a whole other panel discussion about what IQ even means. And I think there's also a kind of a presumption built into that somehow society would be better if everyone was better at math. And, when you really examine that as a premise, I think it's pretty flimsy. So I think, to me, one of the answers to this is just a growing appreciation of the value of diversity in all its many forms, which in a way, I think the CRISPR story, the discovery of CRISPR actually is an amazing example of that. All the different scientists who came together working in kind of, and I mean this in a wonderful sense, kind of a scientific backwater compared to what usually gets funding and attention. This was an area of science that, nobody thought was going to yield a world-changing technology, and none of the people working on it expected this to define their careers or change their careers and, put them in the history books. So it's another reminder of just, having humility about how much we really understand about where things should go or where value in our society really comes from.
Jennifer, there are multiple questions about how CRISPR could be used as a tool to fight cancer.
Well, I think the primary way that it will be used to fight cancer is in concert with another extraordinary technology that's advancing quickly, which is cancer immunotherapy. And so the idea there is to use CRISPR to help program a patient's own immune cells to target tumors. That's something that is, I think, has very exciting future potential where CRISPR could play a big role. I think it's, a lot of people have asked about, can CRISPR be used to correct the mutations that make cells become cancerous in the first place? And the answer there is, in principle, yes, but I think in a practical sense, that would be very hard to use therapeutically, because we would have to get access and correct every single tumor cell in the patient, and I think that's just very, very difficult to do.
But are there edits that could be made, including we read about elephants having particular genes that are particularly useful in fending off cancer, that could make us less susceptible to cancers?
I think that is a fascinating idea. And right now it's really in the realm of future work, future potential. But I think it's a very intriguing possibility that I sort of think about it in my own mind as genetic vaccination. Could we effectively vaccinate ourselves genetically to resist disease that we might get in the future, whether it's something like cancer or cardiovascular disease, or whether it's actually an infectious disease? And I think that's something that CRISPR does open the door to investigating those possibilities.
We only have a few minutes left, 2 or 3 minutes left, so I'm going to ask a final question to both of you, beginning with Adam. And it's the same question, really, because Adam, you've talked quite a bit about the beauty of the basic science that people were pursuing, not knowing they were going to end up with tools that might edit genes or end up with technologies that might even fight coronaviruses, or that even think about things, that people were pursuing this out of curiosity. And your film is so beautiful in that way of just showing that pure curiosity driven science. And that Jennifer has a line I use many times in the book, all attributing it to Jennifer, which is, nature is beautiful that way. I mean, it happens over and over again in the book. There's something, and I say, nature is beautiful that way. That's the theme, really, of your documentary. And I think it's the theme of the whole CRISPR story, too, Jennifer, which is people pursuing basic curiosity about nature, discovered things that then end up being very useful. So I'd like both of you to talk about the beauty of basic science and how that can lead to things unexpectedly that transform our lives. Adam?
Well, I think, I'll just say that for the film, that was something that was so important for us to capture. One of my favorite things that anyone said about the film is that you feel this kind of back and forth between the oh wow and the oh no, the kind of awe of discovery and understanding how nature works, and then the consternation about different dystopian scenarios that might come from this technology. But that awe and wonder and that drive to understand nature is such a fundamental thing about humanity. And I think it's, it's often missing from stories about this kind of science that involves a technology that is also controversial. But I think it's important to remember that it's coming out of a very, very important human impulse to understand.
And Jennifer, final words or thoughts?
Right, I couldn't agree more. I think, for many scientists, we're driven primarily by our curiosity about nature. How do things work? And that's when I think back to my earliest ideas about science and maybe becoming a scientist myself someday. Those were the kinds of that fundamentally was sort of what was driving me, was, I want to understand this. I want to understand the molecules that are involved in this biological process. And I'm still driven by that question. So I do think that the CRISPR story, as we started off with this point in this discussion, is that it's a great example of how that kind of, very fundamental work that isn't targeted in any particular direction beyond understanding a biological system and answering a question can proceed in really unanticipated ways. And that's where a lot of breakthroughs come from.
Nature is beautiful that way. Anne and Rick, thank you so much for having us on. Jennifer and Adam, that was fascinating. Let me turn it back to Anne and Rick. Thank you all.
Thank you all very much. That was
Thank
really
you.
wonderful. And of course, we are reminded, as you've put it many times, Walter, in the course of this discussion, that filmmaking of this kind is exactly the way in which science becomes relevant to a very broad public. And so we thank you, Jennifer, for participating in this film, and Adam, for you making this film, because I think that it really is a service to us all when we can see science expressed, especially beautiful science, in this way. And Walter, thank you for really bringing this conversation together. It was really wonderful. We don't have an event next week. We do invite all of you to go watch the inauguration. We at the National Academy of Sciences have been gifted with several hours off to be able to go watch the inauguration. The National Mall, of course, is off limits, and I don't think any of us would be traveling down in any case, but we will be watching it. We hope you will do that, but we will be back the following week. We're still working on some details, but we're doing another film screening, and so you'll be getting that invitation in your email. And it is a film about the biography of Oliver Sacks. So look for that, and we hope to see you in a week and a half, right?
Yeah, hey everybody, we got 130 questions.
Now.
So sorry that we didn't get to all of them. There were a lot of really interesting ideas and questions being expressed in there. So, but great questions today. Thank you for being such an engaged audience.
Exactly. So we will be back, and we hope you will be too. So in a week and a half.